program to detract from all of the other things going on. We'll run through, and in terms of the format for today, for anyone who's new to the story, we'll give a brief recap and introduction to PYC Therapeutics. We're going to then work our way from an outside-in perspective. So we'll first have a look at what's going on in life sciences generally, with a particular focus on the US because that's where it's really driven from. We'll then look at how that's informing what's happening within the domain in which we play, which is the RNA therapeutic space, more specifically. And from there, we'll transition to PYC within the RNA therapeutic space and a pipeline review. So we'll go program by program, and we'll have a look at what progress has been made since we last spoke to you at the AGM in November. And in particular, what should you look out for in the forward view for the remainder of the year? And here, remember the aspiration that we spoke about at the AGM is that the company is looking to put 3 first-in-class and potentially disease-modifying drugs into clinical development, so into human studies before the end of next year. So it's a very bold ambition. We know through that exactly where we need to be today and also at the end of this year in order to set the platform for success next year and realization of that goal. So we're going to walk through what you should be looking out for as we progress through that. To give you just a sense of the answers to each one of these different topics that we're going to address so you can structure your thinking before we get too deep into it, and for anyone who might have to leave early, PYC makes precision medicines for patients who have no treatment options today. I think most people are familiar with that now. We do it as part of the broader life sciences industry, and it's been a very, very grim time for anyone who's been following the investor updates for the course of the last 18 months at least. It hasn't been a good time within the industry. The macro trend has been very heavily against. And what we're seeing now is that things are plateauing at the bottom. So the Americans describe it as the green shoots. The Europeans say it's not getting any worse. So we will stick to the European camp for now. But at the very least, it looks like the worst of it is over, which is good, actually, from an investor perspective. Not so good if you're an existing investor in PYC, having ridden out the downtime, but certainly good if you're looking at coming into the story in terms of there's only one way it can go from now. The tide is set to rise. It's a question of when, not if. In terms of the RNA therapeutics specifically, and RNA therapeutics for monogenic diseases, we're going to talk a lot today about genetic validation of the targets. This is a particular subset of the industry that we play in, and the macro trend is very strong for us. It's a very, very good time to be an RNA therapeutics company with a delivery technology. We'll have a look a little bit at some of the commercial corporate elements of what's been going on in the industry with a focus on 2 transactions that occurred very late last year in December. And then we're getting into the pipeline review. RP11, I think people have a pretty clear understanding. We are in the regulatory engagement window. We have filed an investigational new drug application with the FDA. We're expecting a response from the FDA in early March. If we are successful there, we are organized and set to go in the execution of a clinical trial. We're currently anticipating first patient enrollment in April. So very, very quickly getting into the clinic, assuming that all goes well on the regulatory pathway. From there, we will largely progress through a single ascending dose study in the RP11 program, which will give us an inferred safety insight on the acute phase, what is likely to happen in the human when our drug is administered to that patient's eye. So it's a very exciting time there. In the second program in ADOA, you're looking roughly a 12-month gap between the first and second programs, which means that we're looking to file an IND in the first half of next year in relation to that program. What happens between now and then is largely a selection of the specific candidate that we are going to progress, so finalization of the drug. Then we go through a fairly well predefined process of manufacturing that drug to a very high standard, so we can go into GLP toxicology studies or the non-human primate monkey toxicology studies that largely form the basis of the IND submission. So the second 12 months is pretty well defined. The action's happening at the front end, finalization of the molecule and release of the data supporting that drug. And then we move to a second target tissue with the third program, a drug for a neurodevelopmental disorder that is also going through some very exciting developments right now. And what we're looking at there is the selection of the chemistry that we are going to use in that drug. We'll talk some more about that in detail because it brings a very nice portfolio diversification element to PYC. We will open up for Q&A at the end of the session, but I'm also going to pause as we finish each one of these subsections and give you a chance to ask questions as we go through. I think many of you are familiar with what the company does now. The company has a platform technology on which it is building precision medicines for patients with severe genetic diseases. And I think the key thing to understand here is we are looking to create life-changing therapies for these patients. So we don't pick diseases where the impact on the patient is mild. We operate in the domain of very severe diseases in these patient lives. We also operate in a very specific space or subdomain of the genetic diseases. We focus in particular on those diseases that are caused by a mutation in a single gene. And we're going to go into the reasons for that very shortly. So if you wind back the clock about 18 months, the industry was very much focused on platform technologies, things that can scale very quickly across multiple indications, because we were in a risk-off phase where the cost of capital was low and people were really looking for blue sky opportunities. And that has changed very drastically. What's happened now is that people are very much more focused at the asset level. So it's lovely for our company that we have a hedge across, yes, we do have an underlying platform technology, but we have now progressed far enough down the definition of the individual assets that are being built on that technology that you can get a very good sense of what is the profile of that particular drug, program, or asset. Going backwards there. PYC's technology takes an existing class of drug and it makes it better. So many of you will be familiar with the work that's been done in the last 30 or 40 years around RNA drugs. RNA drugs have been so incredibly attractive to the industry because they offer precision and potency. So there's been a huge amount of attention that has been devoted to RNA drugs, but they've had a relatively limited profile of success within that 30 to 40 year window. And the reason for that is they have an Achilles heel. Whilst they are very precise and very potent, they don't cross the cell membrane to get inside the cell where their targets reside very well. They have very weak intrinsic cell penetrating properties. And that's a big problem for a drug that is only active when it's present on the inside of a cell. The great development progress that's been made that is creating the strength of this macro trend towards RNA therapies right now is an enabling technology called drug delivery. And this is coming in different shapes and sizes in different forms. Many of you will be familiar with the lipid nanoparticle that has been wrapped around the RNA drug that forms the basis of the COVID vaccine. So the COVID vaccine has really drawn a lot of attention to this concept of facilitated delivery as an enabling technology for RNA therapeutics, but this is something that's been going on for years prior to that. A lot of progress had been made even before the application of the technology in the pandemic. There's something very interesting here in relation to probability of success in the clinic. If you look at the probability of success in the clinic as defined by modality, a naked antisense oligo or RNA drug has the lowest prospect of success of any therapeutic modality. And as you transition to a facilitated delivery RNA therapeutic, you move to a modality with the highest prospect of success in clinical development. This is important, and it's a distinction from the point that we're going to talk about shortly in relation to the validation of the target. Here, we're not talking anything about the disease at all, rather just what is the modality or the nature of the drug that you are using to pursue that target. And we are also operating here in the very lowest risk or highest propensity of success domain within the industry. So that's what PYC's platform technology is, what you see here in pink and orange. It's an enabling technology that carries this beautifully precise and potent class of drug across the cell membrane to enable it to interact with its target. And if you think about the fact that some of these drugs have been successful in entering market and rescuing disease processes, some of them very successful, and we get somewhere around 100 times as much drug inside the cell with the conjugation to a delivery technology. It gives you a sense of the scope of the number of diseases that we are bringing in play now. And that is what is driving the excitement behind these drugs. 4 key elements underpin the company's strategy. We're not going to talk about the first one here because that is the essence of all of it. We're going to treat that as a special standalone proposition. So here, what we'll look at are a faster path to market. This has got 2 components. What we're focused on in the page at the top here is a faster path through clinical development. The prospect of 2 clinical trials and not 3, because these are patients who have no treatment options today, and the imperative or the urgency of getting drugs to these patients quickly. That's terrific. But what you also see, and what is not on the page, is a very much expedited front end for the drug development process. The industry has moved to around about an 18-month timeline from selection of the genetic target to submission of the IND. That's quite incredible. That's compressing what is usually a 5 to 7 year journey of drug discovery down to an 18-month timeframe. PYC is not quite there yet. We've got some improvements at an operating model still to go, but we are really also dealing with the added complexity of the platform technology. So it's adding a benefit to us in clinical development. It's giving us that very highest P success, but it takes a little bit longer in the non-clinical development to get the combination of the two elements of the drug right. From an investor standpoint, the fact that we are pursuing markets that don't have any available therapies for patients is really, really critical, because it makes it very easy to understand. You're not thinking about what's the percentage share, competitive share of the market that the company is going to get. You are much more directing your attention to the question of how quickly are we going to embrace the whole market opportunity. And we see that as a company. We get inbound emails every week from patients with the different indications that we're pursuing, asking to be involved in the clinical trial. Many, many emails, many, many patients. It's a very good indication of just how focused these patient populations are on the creation of a disease-modifying therapy. What you typically see, and I think Spinraza is a very nice example of a naked RNA drug for a rare disease, they hit 85% of the peak market penetration within 18 months of launch of that drug. So they're very, very rapid uptakes once they reach market. And I think everyone is familiar now, or hopefully everyone is getting familiar, these are very expensive drugs. The reason for that is the industry has decided to purposefully incentivize drug makers to focus on rare diseases. Because whilst individually rare, there are 400 million people worldwide who have a rare disease, almost 7% of the population. Collectively, they are very common, and they were a neglected patient population. I think the industry is also aware of the fact that a lot of the innovation in the industry occurs in the context of these rare diseases. And yes, we are going to protect at very high pricing the rights of those drug developers in those specific markets, but the enabling technology is then available to access in the non-orphan indications. You see it very nicely in the context of, again, the COVID vaccinations. So it's important that we get our heads around those. I've been banging on for years about the importance of genetic validation of targets. It was the underpinning of the company's strategy 4 or so years ago when we set about the journey of making our own drugs. And I'm really pleased that the limelight is very much drawn to it right now. There's a reference here that I would strongly encourage all of you to have a look at. It's on YouTube. The Atlas Ventures 2022 Year in Review. It's a great overview. This is one of the smartest venture capital investors in the world talking about what is happening within the industry. And so you'll hear not just the macro, but at around the 30-minute mark, if you want to skip ahead to that window between 30 and 32 minutes, there's a discussion of the power of genetic validation of the targets. And it's brilliant. It's really, really good. I've got the highlight quote here. When you're following nature's lamppost, much, much higher probability of success. There's nothing, that ... is a pause. There's nothing in between those two sentences in the direct script. So this is one of the world's smartest venture capital investors giving you a clue as to what they're looking for in their portfolio companies. And it's the underpinning of our strategy that we have adopted many, many years ago, but there's much more attention coming to it now. And the reason for it is the way that most people value life sciences companies is through a risk-adjusted net present value. You look at a relatively simple calculation of the number of patients you think you're going to reach, remember, undisturbed by a competitive landscape. You multiply that by the median drug pricing and you end up with a very large market. That's great, but it's actually the removal of risk and the probability of ever reaching that market that we are directed to as a company. Are we going to see a clinical efficacy signal? Human safety first and then human efficacy. So that means that that little r that sits in front of the NPV is actually the fundamental driver of the aggregate number that you're getting out of that calculation. So if you're multiplying by industry standard probabilities of success, you're shrinking that number a long way. And if you're adopting the multiple on that probability of success that is associated with a disease caused by a single genetic mutation, the highest form of genetic validation, you are ending up with a very different number at the back end to what you get if you're looking at the industry standard figures. And just to recap, we mentioned it at the outset, but the company's ambition, we're not looking to put one drug into clinical development. We're looking to put 3 drugs into clinical development as part of wave 1 of what the company is about. We are already working on wave 2, and we are turning our minds to the strategy that will underpin wave 3 of these therapies. PYC will look to file an IND to enable a clinical study to progress in 1 new indication every year from this point on, with the exception of next year where we're looking to achieve 2. Very, very bold ambition for the organization, and it's happening in the context of a hyper-dynamic wider industry. Whilst we are playing the game, the goalposts are also being shifted. And we'll have a chat about that shortly as we move to what's going on within the wider industry. But I'm going to pause there just to see if we have any questions that have come through or any in the room. Just 1 second. We can just give you a microphone.
Very basic, Rohan. In your introduction, you talked about the market over the last year or so. I'm presuming that you just mean the financial markets, which have collapsed in our industry. But the enthusiasm for what we're doing, et cetera, is, the green shoots are there and growing and nothing's changed. Is that correct? I haven't missed anything in the industry. That's from...
If you've missed it, I've missed it too. It's my understanding as well. So it's quite strange. I mean, I look at it and you think about how do we assess the value of what we're doing. We're always very much tied to the intrinsic value of what we're doing, which is a function of the number of patients who have the disease that we are pursuing, the pricing per patient, and then the probability of reaching that market. And there's nothing that has changed on any 3 of those variables other than we are getting closer to the market and we are removing risk. So
I can handle the stock market collapsing. We've been there for a long time.
Yes. So it's pretty radical. You look, some of our nearest peers had peak market caps of $3 billion and they've dropped to 300-400 million US dollars. It's been dramatic, really dramatic. Some of it, I mean, you can't look at it at an individual anecdotal case level. It's happened to everybody. The index is down, but that's in the context of each of them generating their own data. So I think it's largely driven by lots of enthusiasm for the sector on the back of what happened in the pandemic. And then a lot of generalists starting to understand that they didn't maybe have the full depth to understand the industry. And then they have moved out en masse at a time where the cost of capital was rising significantly. So companies that are cash flow negative and have a long time before they start generating revenue were punished very badly. Yeah. And so it's very strange. You've got this terrible commercial macro trend, but a very strong macro trend within the life sciences industry towards the RNA therapies. Yes. Yeah. We've got one question, lots on the plate, but how can you afford to do it all? Options, cap raise, investors, deal. Yes, a good question, and the commercials always come up, so we may as well deal with it, get it out of the way up front. If you look at the company's update, the 4C that went into the start of the current quarter, we had $17 million and an anticipated R&D rebate of just over 4. I'm pleased to say that we've received the R&D rebate, so that cash has now combined, and we have $21 million in cash. And we are anticipating roughly around a $10 million R&D rebate for the fiscal year that we're currently in. We're now 8 months through that year, so 8/12 of the way to our entitlement in that regard. So we've got capital reserves, cash and receivables of around $30 million, which is funding for us right now on a net basis of around 12 months. So we've got a 12-month window in which to look at all of those options that you are putting on the table there. And we really need to look down and look at the relative attractions from an existing investor-based perspective in ensuring that we have the access to the capital to do what we want to do. The nice thing about the RNA therapies, and given that we have removed a lot of the risk or shifted the risk from the clinical trial phase, where does that risk go? It comes into the non-clinical window. So it actually means that you can remove a lot of risk in these programs in the early part of development, and that is the much cheaper part of development to turn your attention to. So it fits very nicely for us as a company. The progress that we're making, the anticipated milestones that we've got in the very near term, as in the second quarter, are likely to have a material impact on the way that the outside world views the value of what we do. So it's hopefully a comprehensive answer to what we've got going on there. We've had a little bit of a chat about the trends in the industry. I think one thing that is probably underappreciated is just how dynamic life sciences is. And so if we take this week as an example of different things that are changing in the macro environment, just in the macro, not inside PYC at all. On Tuesday, we woke up to a report out of the University of Oregon suggesting that they had engineered the genome of a monkey to give that monkey retinitis pigmentosa. And that actually the symptoms that that monkey manifested with very closely resembled or recapitulated the human disease process, which is quite an extraordinary development. This is the power of monogenic diseases now going the other way, not probability of success in the clinic. Everyone is trying to shift the gauge on likelihood of success in the clinic into the non-clinical setting. So we have now, not we PYC, but we collectively as humans have made a model of a human disease in the species that most closely resembles a human that manifests as the human disease. Now it's not RP type 11. So it's not directly relevant for PYC in that context, but this is pretty incredible stuff that is occurring now. You can imagine the power of getting all of your non-clinical readouts, effectively all of them, in a single setting. Tox tolerability, dosing interval, amount of drug in the target organ, and whether or not we are correcting the phenotype. You can actually measure many of the same endpoints that you'd be looking at in the human species in the non-human primate. So that's what I mean by the risk is now shifting left. People have cottoned on to the value of these monogenic diseases, not just for the enhanced prospects of success in the clinic, but how can we use that to drive the non-clinical setting to give us an informed answer as to whether the drug is going to work. So that's pretty wild. On Wednesday, a paper was released that suggested that it would be very attractive for a precision therapy, a gene therapy, either a DNA therapy or an RNA therapy, to be directed towards neurodevelopmental disorders. And they called out Phelan-McDermid syndrome as one of the particular examples of a drug that is crying out for a potentially disease-modifying therapy right now. And they gave a very lovely, detailed manuscript of how to design an RNA therapy for Phelan-McDermid syndrome. So very, very close to home in what we're doing. And then this morning, I read that out of the Verdun Institute, a company that is focused on a delivery technology and enabling platform, has just raised seed capital of in the order of $250 million Australian dollars, 193 million US, just to get off the ground. Things are changing all the time. And we need to look at what's going on in the outside world, which parts of it are relevant for what we are doing internally, and how are we going to respond to those. But everybody needs to understand there is a very, very strong desire to change the course of human health. And it's driven largely in America, and it is supported by enormous pools of capital. And this is the field with which we are competing. And we have to remain relevant in that context, keep up to date with what's going on, as well as differentiating between signal and noise, because there's a lot of noise in life sciences as well for those same reasons. So we've got to discern which parts are relevant for PYC, what are we going to do in response to them, but not get carried on with the hype and be very focused in terms of what we're doing internally. So that's just an illustration of the hyperdynamism that's occurring. We mentioned at the outset, from a commercial perspective, it looks like the worst is behind us. And so that's nice. It's just nice to no longer be in free fall. And if you look at a couple of the IPOs that have gone through in the US, they're getting very strong aftermarket performance now. Now, this is very, very small numbers of companies because not many of them are finding the opportunity to list in the US. It's still very, very difficult from a commercial environment. But it looks at least like it's not getting any worse. And so it's a question then of how long it takes for things to turn around. We've got a question here. Press and hold the power button, Karen.
No.
Yeah.
Got away with one silly question. Can I ask another?
Go for it.
Is there, maybe unanswerable, is there any chance at all that something can come out of left field and completely, how can I put it?
Wipe out PYC?
Yes, yes. Make us irrelevant, if you will.
Yeah, you're loath to never say never, but it's highly unlikely. And actually, this is what I meant by we've got to make sense of it. So let's take the delivery technology as an example. If they've got a better delivery technology than PYC, we've got a couple of questions to answer. Is there anything we can learn from that delivery technology that we can incorporate into ours, keep the benefits of our delivery technology, but exploit the benefits of theirs? And if the answer to that question is no, it's a question of, is their delivery technology relevant to the target tissues and cells that we are pursuing? And if the answer to that question is yes, it's whether or not we need to license in that delivery technology to combine with our RNA therapy to create a better drug. So those would be the kind of implications that we'd be looking to discern. Given that we've got this hedge across the delivery technology and the RNA drugs, and we'll come to the conversation later, there's actually a lot of diversification between our different assets. We're not all in on any one of them, and there are sufficient differences between them, both from a biological perspective in the underlying disease, but also the molecules themselves that we're putting forward, that we're not really at risk on any one outcome. It's more 3 independent shots on goal in very attractive markets. So for us, it's much more about making sense of what's going on in the macro and working out whether there's anything that is relevant for us rather than fearing the risk of being wiped out entirely. And I think the other dimension to it that is probably quite important to get ahead of, because the field is moving so fast, we can be a part of those enabling technologies ourselves. So things like the power of the genetic validation can inform changes that we make to the front end to continue to benefit from the macro trend towards the RNA therapy. So I don't think so. I think it's such a good time in the subspace that we're in that there's nothing that's likely to really push us completely out of the competitive landscape. Sorry, one other question that's come through online. Yeah, so we've got a question coming through the chat. You mentioned your drug delivery mechanism and its importance. I see in the literature that there have been issues with AAV carriers in terms of liver and nerve damage. Can you please comment on your carrier and its safety? This is a very good point, and I think it's one of the issues in the competitive landscape that we've been quite focused on, and a reason that we've stayed away from viral delivery vectors. If you look at the literature, not just AAV, but viruses generally carry some very significant safety concerns. Firstly, because they're immunogenic. They elicit an immune response. And secondly, because of the risks of integration with the human genome. Now, AAV specifically is supposed to be what they call a non-integrating delivery technology, but we know that it does in fact integrate. And actually that's the underlying basis in particular of the liver damage that you are referring to here. The extent of the safety concerns go well beyond those. And so the answer in terms of the comparative assessment, because we are using a very, very small fraction of a host complete viral genome as our delivery technology, we don't see any evidence of immunogenicity in the screens that we do in the non-clinical setting, and we haven't seen any evidence of adverse tolerability related to the delivery technology in our GLP safety studies to date. You guys have seen the data that's come out there. So that's the fundamental distinction, is we don't see the same issues with our delivery technology because of the different nature of our delivery technology in that context. The other thing that I think is particularly nice from PYC's perspective is, remember that our drugs, at least the first 2 indications that we're pursuing, we have a localized delivery environment. So we're delivering drug into the eye. And when we look in the non-human primate species as to whether any drug has leaked into the systemic system, into the circulation, the answer is no. It's the presence of the drug is below the limits of quantitation. So if it's there in such small quantities that we're not getting what we call an off-target distribution to the other organs. So the effect is likely to be limited to the eye, which is very encouraging there. All right. Back to the industry trends. A further example here, just of the extent of the difference between what the industry is doing and what those who focus on monogenic diseases are doing. And I think this, again, we've touched on the power of the genetic validation, but I think the last sentence in the first quote here is critical. Oh, sorry, I just got rid of it whilst I'm trying to refer to it. This is a paradigm shift. We're not talking about a subtle change in the probability of success. We are talking about a fundamental change that drives what is a very different lens through which you view the company that is required compared to what goes on in the industry as a whole. And the reason for that, I think you can get your head around what's going on. If you know that this is the specific gene that is causing the disease, and exactly what is happening within that gene, is it expressed too high or too low, that is resulting in the phenotype or the patient's disease process, then you know exactly what you need to do to fix it. It's a very precise thing that needs to be done inside the cell, and we are using a very precise technology with which to do it. That creates a very different paradigm. And I think this point is interesting as well, particularly for investors in PYC as we transition to a combined or phase 1/2 study in humans. The first in human studies, they're a good place in the value chain to be. And I like the quote, it's the CEO of Novartis describing why they are no longer interested in the outcome of phase 2 assets. They don't want to play in the late clinical space anymore because they don't want to buy a drug that's had 90% of the risk removed and benefit in the final 10%. They want to be involved in the journey of the value creation, of getting the human safety and the efficacy signals and enjoying the fundamental re-rate that comes from vastly increasing the P success. And if we are right here, and I think you see increasing evidence, we've given you some references to go back to and have a look at the power of this genetic validation and how far it's changing that P success, you're effectively benefiting from that tailwind in PYC for free based on the valuations that we're attracting relative to peers. So it's the combination of these two that is starting to make things look very different from the investor perspective. All right, any questions on what's going on in the industry as a whole? We don't have any coming through from the chat forum. We've got one in the room, one moment.
Hello. Do you see that there is the opportunity to out-license the transport technology that you have? You see that that's an area of something that the company could achieve?
Yes, that's a good question. And I think that a question in the minds of a lot of investors is, look, if you're talking about this paradigm changing delivery technology, and it's great that you've got a bunch of internal opportunities for its exploitation, but what about out-licensing it, generating some revenue, proving that you're on the right pathway? The answer, unfortunately, is it's a little bit more complex than that, because the nature of the delivery technology is determined by the type of cargo that you are trying to deliver. So you need a very different delivery technology to deliver a pizza to a bale of hay. And so that's the limitation. You really need people who are, firstly, we've chosen to be in the RNA therapeutic space. So all of our data, all of the beautiful validation of our technology occurs with an RNA therapy as a cargo. So that's going to be very attractive to people within the RNA therapeutic space, but not so attractive to people who are operating in a different domain. And then if you look inside the RNA therapy space, most people are working with a backbone chemistry that carries a negative charge. And so if you link up a backbone chemistry that has a negative charge with a positively charged delivery technology, they're going to neutralize one another and prevent either part from operating as it's intended. So you've now ruled out the vast majority of the RNA therapeutic space as well. If you look at those who are leveraging the safety of the morpholino backbone, morpholinos don't have good intrinsic cell penetrating capability. So a lot of these companies have already got their own delivery technology, and they are a genuine like-for-like competitor with PYC, apart from the fact they've chosen different applications to apply their technology. So if you look at a very good example is Sarepta. Lovely proof of concept of the technology, but in conversation with them, it was very clear that if their delivery technology was successful in the clinic, they were not coming back to have another look at getting a better one. Enough is enough. We've got it. We don't want to start the program for a third time. It's only if we run into a big problem in the clinic that we would look to license your technology. And so the interesting thing there was, what are we now cheering for? Them to fail and want to look to do a licensing deal, or for them to succeed and clinically validate our technology? And I think both from a patient perspective and actually from a PYC fundamental perspective, far, far better for us that they went and got the validation of the PPMO technology. And now what you see is others coming into the space. So you see PepGen, they've got their own CPP delivery technology. You now see Entrada, they've got their own CPP delivery technology. So the companies who are in the space now oftentimes have their own modality. Now, you could say, woe is me, we're not doing a BD deal. I think you've got to look at it and say, hang on a minute, everyone's coming in this space because it's a very hot area, and just be happy that actually the value then is in the molecule as a whole and what we can do in the specific indications that we're pursuing. But hopefully it gives you some color as to why BD around the delivery technology itself hasn't been such a focus. We're probably more interested from a BD perspective in the assets as a whole and regional licensing deals, as we've spoken about previously, especially now, because the value of those assets is getting a lot higher. And so that's what you should be looking for is, okay, if this is all true, if we are seeing a very, very strong macro trend to the RNA therapies and facilitated delivery of RNA therapies, are we seeing evidence in the industry of people being willing to cut big checks for early stage assets? That would be the confirmatory proof that we're looking for, right? And if you have a look in just the last month of last year alone, 2 of the companies that we speak about have done big deals at the asset level, not around the delivery technology. One of them doesn't have a delivery tech. That's Wave Therapeutics, licensed for $170 million upfront, a preclinical program that then comes with milestones and a royalty that sits at the back end of that. And that was one of 8 candidates that was part of that deal. So that's $170 million per candidate upfront in cash, US dollars, for a preclinical asset. Yeah, very, very powerful. And that's a naked RNA therapy. So that's a lovely indication. If you look at the commentary around that transaction, that's GlaxoSmithKline trying to get a seat at the table in the RNA therapeutic space. So that was nice to see. But I think even better for us was the Vertex deal with Entrada, $250 million US dollars upfront in cash for an asset that's 12 months behind RP11 or more. Yeah, early preclinical development, and sits in a competitive market space in a rare disease. So that's the 3rd, possibly 4th RNA therapy for a disease called myotonic dystrophy. That was the subject of that transaction. And again, milestones and royalties sitting at the back end. So you are seeing in the industry deals being done that are very much attracted to exactly where we're at right now. And it's for the reasons that we've spoken about. Okay, we've got one more question from online. Are there any plans to collaborate, license out VP001 to pharma as phase 1 completes? This is a good question, and it relates back to the earlier one in relation to sources of capital for the company. The answer is informed by what's happened in the broader capital markets environment. So as the valuation of the parent company has come down, the appetite on the part of the company to look at BD or licensing deals as a source of revenue has gone up. And it should, right? So whilst it's important not to farm ourselves out of the downstream value associated with these assets, particularly because we are not seeing the value of those flow through to the parent company level, the question is, can we get it recognized at the asset level? It would be preferable for us at this point to be entering into the space of doing a transaction of the nature that we've just spoken about. So yes, we are looking to do that. I don't think you necessarily need to be in clinical development. As we've spoken before, preclinical development is a very attractive space for people to be doing transactions. The clinical window, and as soon as you're into a phase 1/2 combination, as you've just seen from the quote from the Nevada CEO, that's a very, very attractive time. That's a window where you're going to get a lot of competitive tension around those assets, which is why we have been so focused on becoming a clinical stage company. So yes, we are looking at the opportunity in particular, again, in relation to regional licensing rights, to retain exposure to the assets in the particularly attractive markets, but to bring on a partner who can join us on the journey and provide additional capital for PYC. Oh, we've got another one that's coming in that window there. Would PYC consider accepting a favourable takeover offer? I think that would depend on the definition of favourable in that context. We look at anything that comes across the table, and if the terms are particularly attractive, we will do what's in the best interest of shareholders. But I think importantly, we're not dependent on anybody else. We're very happy pursuing the progression of our assets towards markets, and we are now in the critical window that is going to inform that value uplift for shareholders. Human safety, human efficacy. It's coming really soon. Okay, so the program deep dives. We just go now one by one. I just have a quick look at time as well, 40 minutes too. quite a long way through. So I might go reasonably quickly through where we've got to and then throw it open to the floor for questions. We won't spend a long time on VP001 because I think it is quite straightforward where we've got to. But just a quick reminder of why we are doing this, who we are doing this for. The patients who are losing 40% of their quality of life because they have a progressive and irreversible blinding eye disease and no treatment options. That's who we're doing it for. There's a significant patient population in Perth. There is a larger patient population on the East Coast. There is an even larger patient population in the Western world. We've spoken before about the size of those markets from a commercial perspective. Looking at the orphan drug pricing here, you can see that as soon as you hit a patient population of around 7,000, you're into the $1 billion plus market space, which is small by industry standards, but very, very attractive in the context of first-in-class assets with a single genetic underlying cause. I won't go through the other dimensions there. Where is this asset heading and what you should be looking for in 2023? I think we know the short-term objective is the clearance from the regulator that we have done everything necessary to support the proposal in relation to the clinical trial design that we've put forward. And if we are successful there, we'll progress through what they call a single ascending dose study. And so what that means is we will start with a low dose. We will dose 3 patients with the drug and we'll wait a time period of 4 weeks to ensure there are no acute tolerability issues. We will then hold a meeting of our data safety committee and we'll request approval to progress the dose escalation to a mid dose. And we'll dose another 3 patients and we'll wait 4 weeks and we'll take the data on the acute tolerability back to the safety monitoring committee and we will ask for approval to escalate to the high dose. And once that's done, we'll follow those patients for a period of 24 weeks to make sure that we're looking at the chronic tolerability window as well. And then we will convert to a multi-dose study. We'll take likely 2 of the dosing cohorts and a 3rd group of patients who are not aware that they're not receiving treatment, but in fact are not receiving treatment. So that will give us a placebo control. And that will then be part 2 of the study. What the results of that study, plus the natural history study that is already underway, will inform. If we look at the natural history study, what you're seeing there is how quickly does this disease progress in the absence of treatment. And what you'll be comparing that to is how quickly does this disease progress in the context of treatment. And from the comparison between those 2 studies, that will then inform how many patients we need to recruit into the pivotal study, the registrational study, and what the design of that study looks like depending on the endpoints against which we're seeing progression or otherwise of the disease. And that will then form a conversation with the regulator to make sure they are happy with that for the new drug approval application that sits at the back end of it. And that will be how we design the pivotal study on VP001's path to market, assuming that we are successful. Please feel free to send any questions through. Any questions on RP11?
Well, am I right in assuming that the use of this, where you're developing, will prevent degradation in sight, or will it improve the sight of those that already have lost some of their sight?
It's a very good question. I think the answer is complex, unfortunately, but I think it's safer to start with the latter element of what you've just described there, which is arresting progression of the disease. Now, in RP11 specifically, and again, this is part of the strategy from several years ago, that is a monumental achievement because this is a slowly progressive disease. So whatever visual function the patient has at the time they start treatment, we would be looking to preserve that. And the interesting thing here is if you look at the patient population, present with the disease in early childhood, progress through to legal blindness in the 4th or 5th decade of life, but not completely blind till the 6th or 7th decade. That means that it should be a very, very high conversion ratio of the addressable patient population to the eligible treatment population. So we're going to get nearly all the patients, if not all, which is very encouraging. And that if we can preserve the vision that they've got, even if you get a phase shift to the right and you give people the functional vision above legal blindness threshold for an additional decade, 2 decades, this would be life-changing for those patients. The really intriguing thing is that in the gene therapy for another form of RP, they actually saw an improvement in the patient's vision. And the posited reason for this, that no one is able to prove at this point, The theory is there are some cells in the retina that are sick enough to have opted out of processing the visual signal. They're so unwell that they can't function, but they're not yet dead. And so if you can reach them with the drug and restore the expression of the gene that's missing, and you give them time to recover their health, maybe they opt back in to assistance in the processing of the visual signal. And if that's correct, we may well hope to see the improvement that was seen in the context of the gene therapy for RPE65, which would be incredible. Okay, we've got some questions coming through online now. What are your endpoints for the multi-ascending dose? That's a very good question. And so we'll talk generally about the approach to the endpoints at this point has been to cover the field. The first thing that we've got to do is make sure we've got all of the relevant endpoints to the disease process so that we don't miss the efficacy signal. And then what we've been doing is working with clinicians and experts from around the world who are particularly interested in RP11. And we are looking at, and we're actually the beneficiaries of a group in Norway out of the University of Oslo who've done some lovely work on a natural history study in RP11 patients already. And so what we're working with those guys on is looking at which are the endpoints in their natural history study. Have we seen progression of the decline in the patient's visual function to give us an early clue in the phase 1/2 trial of the endpoints that we need to be paying particular attention to, as well as the patient stratification. Which are the patients that are progressing most rapidly on the loss of the visual field curve? So the distinction, the potential distinction between treated and untreated is the greatest. And what we're seeing there is perimetry is the key endpoint at this point. That's the insight that we're getting right now. We are looking across a range of other endpoints, including the visual navigation course that was used in the support of the registration of the existing gene therapy for retinitis pigmentosa, as well as a range of other visual field assessments, including things like the ERG, the electroretinogram, or the retina's response to a visual impulse, looking at the retina using a bunch of tools that are capable of being put on the front of the eye and seeing any anatomical changes, differences in the thickness of different retinal layers. We're looking at those as well as a standard battery of tests that are related to visual acuity. How well can the patient see? And as you guys are probably familiar, having done an assessment on the Snellen's chart or reading the letters, the visual acuity is not likely to be particularly helpful for PYC because central vision is preserved. So that's an endpoint of less relevance. The other dimension that is increasingly getting recognized and where we're a beneficiary of the work that's been done by others, patient-reported outcomes. So a questionnaire that the patient fills in, reporting on whether or not they are in their functional activities of day-to-day life, noticing an improvement in their vision. So that's a high-level overview, and thanks for the clarification. That's on the efficacy side of things. Can we please clarify the timing of the commencement of the Part 1 and Part 2 trials? So the usual caveat of pending acceptance by the US regulator of the IND that's been submitted, will be looking to start the clinical trial in Q2. That's Part 1. And then the timing of the transition from Part 1 to Part 2 is informed by the efficiency of our progression through the patients in the single ascending dose study. So if we can get through, identify the patients and treat the patients, the 3 in each group in the single ascending dose study, we only need to finish the dose escalation and then wait 24 weeks before we'd be converting to Part 2 or the multiple dose study. The real advantage that we've got here is that we've got patients who are already enrolled in the natural history study who we can convert across to the interventional study if we want to ensure that the enrollment of the patients in the interventional study is efficient. So we should have control of that. High-level top-down answer, the safety study is this year. We're moving to the multi-dose study next year. I've got them, Carol, up here. Yeah, so I've got the best case when would this drug reach market? And then could the phase 1/2, Part 1 and Part 2 overlap at all? So it's interesting. It will answer the second one first, not the Part 1 and the Part 2. No, you need to establish the maximum tolerated dose before you move to the multi-dose study. But the Part 2 and the pivotal could overlap. Yes, there's the potential for that to happen. And so that then informs a complexity, Robert, that means we can't answer your question entirely at the minute because the design of the pivotal and when we initiate the pivotal and whether it's overlapped with the Part 2 will be dependent upon what we see in the early phases of evaluation. But what we can say is that the FDA requires one 24-month study on safety and one 12-month study from an efficacy standpoint. in order to support the registration of the drug. So it gives you a sense of the critical path timeline. Most important is for people to understand that it's not so much the timing of the entry of market, it's the timing of the establishment of the efficacy signal that is likely to be driving the valuation of the company. Okay, so we've dealt with those ones. We've had a good go at RP11. ADOA is a very, very exciting program. And there's lots happening right now in this program as we move to confirm the final candidate that we will then be sending off to the drug manufacturer to make in very high purity for us to enter those toxicology studies that we need to be into in the second part of this year in order to be prepared for the IND that comes next year. There is, firstly, before we get to the quote on the page here, we continue to invest in our platform technology. And we are live at the minute in a study that is evaluating a second generation delivery technology. The early indications from the small animal, from the mice, is that we have a delivery technology that is much more potent than the delivery technology used in RP11. This will be confirmed very soon in the context of an eye that anatomically is closer to a human's, a rabbit. And if we see a much more potent delivery profile in the target cellular layer, that is going to be a very, very important piece of information, because remember the two questions that you are most interested in. Can I deliver enough drug inside the target cell of interest? Can I modulate the gene that is causing the disease once I'm inside that cell to rescue the disease process? So that's going on the delivery platform side. On the assessment of the drug itself, we're getting into the patient-derived models. So we're now looking at what's going on in control of gene expression in the target cell that is driving this disease process in cells that have been generated from patients with this disease. And you just see a lovely quote here. Again, remember, when I say the goalposts in the industry are shifting, everyone wants to shift risk left in the curve. And one of the revolutionary ways in which this has been done most recently is through the creation of organ-in-a-dish models, the ability to build a human eye outside of a human. It's a pretty radical concept, but it gives you the precise genetic background against which you want to test the efficacy of your drug. These results are coming in Q2 for the ADOA program, and they will be used for the selection of the final candidate there. That gives you a lot of insight, lots and lots of insight in terms of the prospectivity of that candidate's success in clinical development. Remember, this is a bigger patient population than RP11, roughly twice the size. In the third program, this is a hot field at the minute. As we mentioned before, journal articles calling for potential disease-modifying drugs in the context of these neurodevelopmental disorders, and calling out SHANK3 or Phelan-McDermid syndrome specifically as one where the patient population is really going to benefit from a disease-modifying therapy. What we are doing here, and this is one for the more advanced understanders of the PYC story, we're using a naked antisense oligo with a different chemistry to that which we're using in the earlier two programs. And you might ask, hang on a minute, why are we doing that? If we know that the problem with the RNA therapies is that they don't get into the target cell well enough, why would we make the retrograde step of moving back to a naked antisense oligonucleotide? The reason for it is there is a very clear clinical and commercial validated pathway for a naked RNA therapy for the treatment of a disease of neurons specifically. And there's a beautiful set of data that's come into the public domain from other companies that have been active in the space in different diseases, linking the biodistribution or the profile of where your drug goes when you administer it in a rat to a non-human primate. So that means you can actually get very good insights on whether your drug can reach the target cell in a human from a rat model, which is quite incredible. So what we're going to do is we're going to use the rat model to go head to head with what the field are doing with a naked RNA therapy, which we own through the composition of matter patent that we've claimed, head to head with our facilitated delivery technology, the PPMO. We've got a choice between the two different technologies, one that's already validated and one that we think is better, but is yet to be fully validated. And we will leverage into the studies that have been done linking the rats to the non-human primates to show the outperformance of our technology in that context. It is quite possible in doing that, if we see what we hope to see, which is a much greater biodistribution of our PPMO as compared to the naked oligo to the neurons, that we will establish a new delivery paradigm for RNA therapies in the CNS in the process. And that would be quite powerful for us. The difference there, and the reason that we haven't jumped straight into it from the eye to the CNS, remember the eye is a very small organ. And the proximity of the tip of the needle when you're administering the drug to the target cells is very, very close, in the order of a centimetre. This is not the case in the central nervous system. The drug is administered to the base of the spine. It has to traffic through the cerebrospinal fluid all the way up the spine inside the skull and then distribute within the brain. It's a more complex route of administration and target tissue that we're dealing with, hence the desire to stick with the proven and only knock it off once we've got definitive evidence that we are better than that technology. So very exciting in Phelan-McDermid as well. We've also started the early work on the Wave 2 candidates, picking the indications that we're going after to support the medium-term aspiration of the company. Very good that we have the aspiration to put three candidates into clinical development in the next 24 months. What comes next? Given the strength of the macro trend towards this field, given that we have 7,000 monogenic diseases, 95% of which have no treatment options available today, we are scaling up in a big way. And so look also for additional population of the pipeline with the candidates that are coming down the pike in the longer term. The reason that we're doing that, the reason we are scaling our technology so quickly, is because of the differentiation on the delivery technology, coupled with the standardization in the chemistry of the RNA therapy itself. And what that enables you to do, once you've set those two things, you can really move quickly, really, really fast. So we are leveraging now the synergies that we've captured through the comprehensive non-clinical evaluation of the RP11 candidate into other programs. And we're going to use that to drive very fast in the scalability of our technology, because whilst the industry is on assets and off platforms now, they'll be coming back for platforms at a time when things are getting better, the cost of capital is cheaper, the sentiment is higher. What we want to have at that point in time is not just a pipeline of a lot of assets that are attractive for the reasons they are right now, but also have demonstrated as much as we can the applicability of our platform technology to target tissues, target indications that go beyond the eye. Those rat studies in Phelan-McDermid are also being conducted now, and we're expecting the results of those to start coming out in Q2 as well. So hopefully I've given you some sense of the scale of the work that is occurring within PYC today. I hope you also have got some sense there is real palpable excitement right now. If you think about what are the actual implications, what's the so what of everything that we've spoken about? Well, we are a company that sits in an industry that is going headlong towards RNA therapies. And we are playing in a space where we've got not only the highest probability of success by modality in the facilitated delivery of an RNA tech, but also the highest probability of success by indication, genetic validation of the targets that we're going after. We're doing that with a rapid front end, an 18-month timeline from nomination of a target to submission of an IND, at a time when we are simultaneously generating human safety and efficacy data, the currency of the industry right now, in our more advanced candidates, in the context of a faster path to market, a captive patient population sitting at the back end of it, and very high drug pricing. It is a really, really good time to be an RNA therapeutics company, particularly one with a facilitated delivery technology as well. There is going to be an enormous amount of patient impact that comes from these technologies over the coming 10 and 20 years. Very, very significant returns are going to be made within that window. The space is right. The question for the investors is, can I pick the right players within the industry? And there is a lot to say for PYC's approach and where we sit right now. Hopefully, we've been able to give you some sense of what that looks like. It's going to be a very, very exciting time in the immediate future before the end of this fiscal year in the first half for PYC shareholders. So hold on to your hats. I'm sure it'll be a white knuckle ride. It has been so far. It will continue to be so, but a very, very exciting time for investors, and one that I hope is going to be very rewarding. All right, we'll open up the floor again. We might take, I think we've got an additional one online. Could we describe the development of the lab and staff over the last few years to achieve all of this? How many are there, etc.? I think this is a really good question. And firstly, people are an asset in that context. We haven't spoken about it now. It's not yet ready for prime time, but there's a bunch of other stuff going on in the background that is driven by extraordinary scientists from within the team, having learnt a lot in that time frame and having a captive pool of these patient-derived models already up and running. So we can evaluate changes in different applications of RNA technologies and make sure that we are moving with the space in that context as well. That's driven by our people. Our people in the discovery team in Perth, we have 60 staff here in the discovery operations, and we're complemented by another 4 in San Francisco, who focus more on the development side of things as well. One of the key things that we're missing in the Australian ecosystem is the industry experience of knowing what the minimum viable product is. There's 100 beautiful things that we could do in evaluating these drugs in the non-clinical setting. What are the 10 that we really need to do that give you the 80-20 answer, that enable us to move quickly? And that industry knowledge has been brought on through the introduction of those key staff in San Francisco. And we are really working as one team. We're integrated now in moving these assets quickly. So I'm very pleased with the evolution of the industry, the evolution of the company within the context of a hyperdynamic and moving industry. We've got to keep doing that. There is a lot going on in, you all have seen what's happening in artificial intelligence, analytics, RNA technologies. We've got to evolve, continue to evolve the structure of our discovery and development operations to make sure that we are keeping up with what's happening at the field. So it's not just about the assets, it's about how we're evolving the company for what's going to come next as well. I think relating to the question earlier around competitive threats. Okay, any other questions from in the room? Please feel free, if you've got any online, to send them through. Anything from anyone else? No takers? We'll give you a couple of minutes online just to see whether or not there's anything else that wants to be asked. Oh, sorry, we do have one that I skipped over. What will be the visual requirements for the patients in the phase 1? So this question relates to the points that I was talking about earlier. In terms of the visual requirements, we don't have any specific criteria for the patients from an eligibility perspective. What I think we're more interested in is whether or not we can stratify patients by the stage of their progression within RP to ensure that they are going to give us an opportunity to look beyond the primary objective of the study, which is safety tolerability, to glean some insight on what is happening from an efficacy perspective. So we are going to be looking for and trying to generate some early readouts in relation to the efficacy side of things. And for that, as I mentioned previously, we need patients who are progressing on the different endpoints that we're using in the clinical trial at a rate that is going to enable us to see a deflection in the context of treatment within the time frame that we've got in the clinical trial. This is the flip side of it being a slowly progressive disease. It takes a while to see the delta. So there's always 2 sides of the coin. The benefit, if we get through, we capture all the patients. The downside, it could take a little bit longer in order to see a significant enough difference between treated and untreated in order to know that the drug is working. So there are none from a requirements perspective. It's much more about the thinking that we do in terms of selection of the patients through that stratification process that I've spoken about. Some more coming through. Are we able to talk to the company that has raised $1 billion? I'm not entirely sure which company you're referring to there, the $1 billion. There was $250 million raised by the delivery technology company overnight, Peter. This is standard fare for the US. This is happening daily. So we're not restrained in terms of who we talk to, but we're not looking for an enhanced delivery technology right now. We're very, very happy with the results that we've seen to date. And I think we're very keenly anticipating the results of those rabbit studies that I mentioned are being conducted in the platform context to have a look at how far can we improve our own delivery technology. I guess the question would be more, do they want to talk to us, given that they're a delivery technology and not an RNA drug design entity, whether or not we could partner from that perspective. Really what we do there is we take BD as a whole. We try and communicate very clearly what we do, and we share that with people who have an avowed interest in the areas of overlap, common interest. And that's what we look to do from a business development perspective. But we've got discussions ongoing at all stages of the chain in that regard, and we'll continue to do so. A question coming through, are we concerned that more rapidly progressing patients could also be more difficult to treat? Yeah, that's a very good question. And so firstly, it's the reason that we went with RP, not more severe phenotype. So we had a look at conditions, and for those who've been committed, rusted on shareholders to PYC for a few years, remember going back to the strategy considerations around why we steered clear of the Leber congenital amaurosis and the concerns that we had with the KEP290 programs that were going on in that regard. When you talk to the clinicians, these patients have not many viable photoreceptors at the time they're born, and they fall off a cliff in terms of the loss of those photoreceptors. So yes, we definitely are concerned by that. We've mainly addressed it in the context of the strategy level by picking an indication where you tend not to see patients who are progressing rapidly. And so what you see in the natural history studies that have been done, and it's Josephine Prenner-Holton's group at the University of Oslo, it's some very elegant work, is that the maximal rate of progression in RP11 is a loss of around 8% of visual function a year. And there aren't many, if any, I don't think I've seen any outliers who are progressing at a significantly increased rate as compared to that. And you see preservation of the visual function almost uniformly in the patients through to the later decades in life, fourth decade onwards. So yes, we would be worried about it if it was a feature of RP, but we are not, because of the slowly progressive nature of it and what's been observed in the natural history studies to date. Last chance. Maybe whilst we're waiting, giving people a final opportunity. So there were 3 things that we wanted to do to enable you to ask questions and to become a bit more engaged in what's going on within the organisation. Hopefully we'll fulfil that one. Secondly, to share the enthusiasm and the excitement. Sometimes I feel a bit like a used car salesman, getting too excited about what's going on in the company and being evangelistic for what's happening. But we don't want to go overboard. It's a pretty tense time. It's a hyperdynamic industry. There's lots going on. We've got to respond to it all. But I think we are in a very, very strong position now. Hopefully the organisation can really attack the upside of that, especially if we are to be supported by a rising tide from a macro sentiment perspective. That would be lovely. But the one thing that I did want to do today, in addition to enforcing the messaging and encouraging you to go and read those resources in relation to the power of genetic validation, it really is the central premise to the PYC investment proposition, is to give you a sense of what's going on outside of the RP11 program. There is a lot happening and it's coming to fruition in the very near term. So stick with us. I think we're going to give you a lot of insight into exactly where we're at on the programs 2 and programs 3, as well as the platform itself and the next wave of therapies that are coming. And you should continue to think about those 2 core questions as you look at and evaluate the communications that we're putting out to market. Can we reach the target cell? Can we modulate the gene expression profile once we get there? If you can do those 2 things, you really do open up a whole new paradigm in terms of what you can do to rescue a disease process. 2 more snuck in. Can you comment on the following? Well, that's a long question. One of the main risks associated with RNA-based monogenic therapies is the potential for off-target effects. RNA molecules can interact with unintended genes, leading to unintended changes in gene expression and potentially harmful side effects. To mitigate this risk, researchers must carefully design and test their RNA therapies to ensure they target only the intended gene and do not cause unintended effects. Looks like we might have had copy and paste from a scientific paper going on here. Another risk is related to the delivery of the RNA molecules into the patient cells. The RNA molecules used in these therapies are often fragile and can be degraded or eliminated by the body's natural defenses, making it difficult to deliver them to the targeted cells. There's also the possibility of the immune system reacting to the RNA molecules, which can lead to inflammation and potentially harmful immune responses. So yes, and there are answers to each one of those elements that inform the specific selection of what PYC is doing. So this is good, but what looks like a paper is referring to RNA therapies as a whole, which includes messenger RNA therapies, which are much, much longer pieces of RNA than what we're dealing with in the context of antisense oligonucleotides. So the shorter the sequence that you're using, the lesser the risk of the immunogenic potential. And remember that we also have the ability to evaluate immunogenicity before we get into a human. So we take human, we go to the Red Cross and we get human mononuclear blood cells, and we look at the immune response, the inflammatory response in relation to the administration of the drug in that context, which gives us a lot of insight into what's going to happen when we get into the human. Coupled with that is that we look in the most sensitive species, rabbits, from an ocular perspective in our toxicology testing, and we look in the most toxicologically relevant species, non-human primates. So we're evaluating in both, and this is something that I think is important actually. The GLP toxicology studies have been conducted at 3 different doses, and the NOAL, or the no observable adverse event level, so the dose at which we see nothing bad happening, is the highest dose that was assessed. That's 50 micrograms in a non-human primate. If you scale the size of the non-human primate eye to a human eye, you're looking at an adjustment factor of between 2 and 2.5. That would suggest a human dosing of between 100 and 150 micrograms is safe. We are not looking to exceed that dose. We want to dose less than 100 micrograms in a human. So we get a very good insight into the likely tolerability profile on the back of those studies. In terms of the RNA molecules and their degradation, again, that's much more of an issue for the mRNA than it is for the antisense oligonucleotides, particularly those that have been stabilized with the morpholino backbone chemistry. They're not subject to degradation. And the reason that we're confident here that we're getting the right outcome is because we also look at target engagement in vivo. So we're actually looking not just at how much of the drug or component parts of the drug are recoverable from the retina of the rabbits and the monkeys, but also looking at whether the RNA therapy has actually got inside the cell and engaged with its target. So that's giving you an indication that it has not degraded on its journey to the target inside the cell. So yes, we can get over that by looking at what we call the PD readout as well as the PK. So PK tells you how much drug is present in the target tissue or the target cell. PD tells you whether or not that drug is engaging with its target specifically. And to have a look at the unintended off targets of an RNA therapy, remember here we benefit from the greater length of our oligos than those that are used by the majority of the field. The shorter your sequence, the greater the risk that you've got homology or commonality with another area of the genome. So the companies that are generating naked antisense oligos are shrinking the number of letters in the sequence down to around 16 or 17. They need to make them small because they're quite toxic when you increase the length of the letters, because the backbone chemistry itself is quite toxic, the negatively charged backbone that we spoke about. PYC's oligos are around 25 letters long. So because we've got the longer sequence, we've got the greater specificity for our target. There's a higher chance that you get overlap just to the target gene and not to any other genes. So that's been very helpful in that context for us. We can use computer programs to what we call blast the genome or check whether there are any other regions that are likely to have overlap with the drug that we've designed. And that's part of our drug selection process that we're going through in ADOA right now. So yes, we've got a lot of advantages in the context of overcoming those challenges that you've articulated. All right. Use GPT chat. Very nice, Neil. That's embracing technology. We firmly advocate for that within the company. Taking on what you just said, what will that dose be sufficient to do what you intend to do? That's a good question, Sean. That's complex as well, because the traditional, what we call PK-PD relationship of small molecule drugs is distorted in RNA therapies. The best indication of whether the drug is going to be successful for an RNA therapy is the concentration of the drug in the target tissue. And we are the beneficiaries of a number of companies that have gone before us here. What we see when we evaluate the concentration of PYC's drug, VP001, in target tissue, is that we are significantly higher than what we see in our peer companies in the RNA therapeutic space. So that is extremely encouraging, because you have not yet looked fully at the power of the delivery technology there. You're looking at how much drug is recoverable from within the target tissue, but that could be inside the cell or it could be outside the cell. So we are comparing against naked RNA therapies that should be there in much higher concentrations, and including RNA therapies that are proven effective in the clinic. And we are seeing a higher concentration of the PPMO or PYC's RNA technology. So that is very, very encouraging in relation to answering the imponderable element of your question, which is, will that be sufficient to do what we intend? In particular there, SRP-5051 from Sarepta, the PPMO that is in a phase 3 study and has already demonstrated efficacy over and above the naked antisense oligo that is already approved for that indication, is present in muscle at a substantially lower concentration than VP001 is present in retina. Very, very encouraging. Remember also in RP11, there are patients who have the mutation but don't have the disease, suggesting a very low disease correction threshold. That combination looks very powerful right now. Okay, I think we got, oh, thank you from Sean. Perfect. All right, very good. Thanks very much, everyone, for your engagement. As we said, it's going to be an exciting time going forward, and we look forward to updating you again in person at the Q2 update.